CU asked, for ROS1 positive patients whose cancer cells develop resistance to first-line treatment of sortitinib, what are the treatment options for such patients? And in particular, are there any new or potentially new targeted therapy or immunotherapy drugs which can be used for patients after first-line stops working?
Yeah, that's a great question. It's interesting that sortitinib is the drug chosen. So sortitinib is not formally approved for ROS1, but it does have activity. The only quotes FDA approved drug is crizotinib. Sortitinib has attraction, however, because it has much better penetration into the brain. So for patients that have brain mets, sortitinib has been approved for a while for ALK-positive tumors. And as long as you can get it, that certainly it was a good option, presumably, for you. Now, what do you do when it progresses? Well, as I just said, there's really no other ROS-approved drugs, but there are drugs that do have activity against that ROS active oncogene. The most recent one is a drug. So I guess I need to make a disclaimer here that we'll be talking about not FDA-approved indication here, right? Just to be clear. So the most recent one, just late last year, is a drug called lorlatinib, yeah, and it was approved for ALK-positive cancers, but it's known that it has pretty good activity against ROS as well. Now, we just participated and completed enrolling to a trial of lorlatinib in both ALK and ROS-positive cancers. The data are still sort of pending and cooking for that. But that being said, I think lorlatinib is a reasonably attractive option. In addition, there are other not yet approved drugs in clinical trial that are of great interest. Chemotherapy, if it hasn't been done before, is certainly an option. You mentioned immunotherapy, not so much, right? So the data for immunotherapy in these molecularly altered cancers is sort of disappointing. And it's not entirely clear why. It may be that the mutation burden is low. It may be that they are immunologically privileged tumors, that are able to, through other mechanisms, maybe other than PD-L1, avoid recognition by the immune system. Not entirely clear. And then the final thing I'd say is the most relevant, if possible, would be participation in a clinical trial, I would say.